talking about the dynamics of influenza viruses, public health, and I will be starting a tenure track assistant professor position in the Department of Ecology and Evolution at the University of Chicago. So all good models are motivated by good questions, and on this slide I've tossed up some of what I think are the most outstanding questions about flu. And I mean outstanding in that I think they're really interesting and they're important from a public health perspective. And they're also outstanding because we don't have answers for most of them, or we have very incomplete answers. So these are also areas where modeling can hopefully inform us. So one of the major questions that people think about from year to year is how is it that we can predict which flu strain will be dominating in the next flu season? And then when will the peak occur? Is it going to occur in December, January, February? And how big is it going to be? And then more generally, we can think about whether it's a reasonable goal to be discussing the possibility of eradication of influenza in general. This would be just in humans. And whether we should think about it in terms of eradicating seasonal flu or just potentially eradicating an emerging subtype. And then on that subject, should we worry about the emergence of H5N1 or more recently H7N9? And how is it that they might develop the ability to transmit easily between humans? So these themes have been introduced, I think, by all of the speakers, but just reminding you, why should we model at all? There are several different purposes for modeling. The first, of course, is to build intuition. If we start with a reasonable set of assumptions about what we think is going on in the real world and we put them together, what kinds of dynamics do we see? Does it agree with our intuition? And most of the time we're modeling because our intuition is very poor. And then another major function of modeling is, of course, to test hypotheses. And so, for example, we might say, does a model that contain seasonally varying transmission rates or some sort of temporary non-specific immune response or rare mutations that have very large beneficial effects, do models that contain these elements significantly outperform models that do not contain those elements? So this is, again, hypothesis testing. And I put significantly in bold because we do use information criteria, and there are a lot of formal methods by which we can compete models against each other. So this is another important function of modeling. And then finally, and this has also been, I think, mentioned by all of the speakers so far, a goal of modeling, an ultimate goal is to predict. And this is actually, it's a separate step from the hypothesis testing because we found that in theoretical ecology and then also in epidemiology, sometimes you can get models that are quite predictive, but they aren't necessarily based on our understanding of reality. So you can use machine learning methods often to develop models that perform very well, but for reasons that appear to be completely unrelated to the underlying mechanisms. And this is a little distressing, but really, really interesting, and it's a fun area of study. So with influenza, there are a few major themes so far in modeling. The first is that, and this is a lot like HIV, we have this convergence of ecological and evolutionary timescales. And that's kind of a buzzword, but it basically means, or buzz phrase, but it basically means that during the course of an epidemic, while we're modeling the population dynamics of the virus, or you can think of it instead as the population of infecteds, we also often have evolution that then modifies these dynamics. And so often models to be effective, but this is of course something that is investigated all the time and there are a lot of different correct answers, but it means that often we have to include an evolutionary component as well as an epidemiological or ecological component. And these evolutionary components can be really, really challenging because often there's epistasis, so there's interactions between different parts of the genome of the virus. Of course, there's often chance events in evolution, and we're not sure how important they are. And then there's a lot of uncertainty, for example. If an amino acid substitution occurs in one protein, is it going to have an important effect on the function or the fitness of the pathogen? And then similarly, there are some really big challenges, and I'll be giving you examples of all of this. There are challenges for the ecological components in that we are often dealing with systems that are chaotic. So they're very sensitive to initial conditions, and it can be generally hard to infer what's going on. We also have noise, again, as with the evolutionary components, and then identifiability, in that there can be many interacting parts, and we might observe only a few of them, and then that means we can't really say what's going on with the parts that we're not observing so well. Okay, so before I get into vivid examples of models that have these problems, and then also some successes with flu, I'm just going to review the biology very briefly. So flu is a segmented virus. It has 8 gene segments. It has 2 major proteins on the surface. hemagglutinin and neuraminidase. The hemagglutinin is abbreviated HA and the neuraminidase NA, and the hemagglutinin number is denoted by what follows the H, and then the neuraminidase number follows the N, and the standard subtype names. So the hemagglutinin is what's responsible, or it's the protein that allows the virus to bind to the host cell receptor. So it performs a very, very critical function, and many of the most effective antibodies that we develop are ones that interfere with this ability to bind to the host cell receptor. We also develop antibodies to neuraminidase, and we also develop antibodies that can be quite effective in neutralizing the virus that do not actually block receptor binding. But for most of this talk, you can just think about antibodies binding to the surface of the hemagglutinin. So this is to give you a sense of how diverse the hemagglutinin is. There are 3 types of influenza, A, B, and C. B and C have been circulating in humans for a very long time, and they do not have subtypes. So that's, they're down here. And then within influenza A, we have these subtypes. This red is, this is from a paper on a recently discovered flu from bats. So there are now 17 different kinds of hemagglutinin. And this is just another depiction of their evolutionary relationships. They are often separated into 2 groups, group 1 and group 2. And because most of the hemagglutinin is actually like the stalk, the 2 groups are mostly distinguished by how similar their stalks are to each other. So with neuraminidase, you also see this evolutionary divergence, and there are, within influenza A, many different types of neuraminidase. What's interesting is you don't see this sort of structure within the internal genes. And I think Don gave you a lot of this information. So influenza A is very cosmopolitan. It mostly infects birds, and a few lineages have established endemic circulation in humans, and I've denoted them here with like the little pictures. So H1, H2, and H3 have at some time or other establish themselves in humans, and then also a few other hosts, and I put those up there too. And types B and C, as I said, have only been found in humans so far. So influenza A has a really interesting history in people. You're all probably familiar with the 1918 pandemic of H1N1. In 1957, H2N2 replaced H1N1. 1968, H3N2 replaced H2N2. H1N1 was reintroduced in 1977, and of course, it was replaced by a new variant of H1N1 in 2009. And again, throughout this period, we've had influenza B and C. And something that's useful to think about when we're discussing the emergence of new subtypes and the replacement of old subtypes is actually what is it that's new and what's really being replaced. So we don't really know for H1N1 how many of the genes were introduced for the first time into humans. Clearly, the hemagglutinin and the neuraminidase were. But with H2N2, there was reassortment. And so the new genes were actually just the hemagglutinin, neuraminidase, and then one of the polymerase genes, PB1. And then with H3N2, we kept the old neuraminidase, and we got a new hemagglutinin. But then with the recent 2009 H1N1, that was all new. And the details are not important here, but this is basically showing you the evolutionary history of the 8 gene segments in the H1N1 virus that emerged again in 2009. So those gene segments are shown here at the far right. So we're looking at this as time in years. And what you see here is, again, the evolutionary history of these gene segments, but separated by host of the ancestral viruses. So birds are the green rows on top. And then viruses from pigs are the orange area in the middle, and then these are viruses from humans. And so you can see that 2 of the gene segments came from birds, and most of the others came from pigs, but some of the ones that came from pigs had previously been in the H3N2 that had been circulating in humans. So this is just to give you a sense of, again, the sheer amount of reassortment that goes on in influenza across different hosts. So getting on to models of influenza. I'm going to focus on seasonal influenza and specifically on H3N2. So this is one of the monomers for the hemagglutinin of H3N2, and the colored areas are thought here to be the epitopes, or the regions that antibodies bind to that are under a strong degree of positive selection. So basically, the hemagglutinin mutates in order to shake off antibodies, to become unrecognizable to prevailing antibodies. And so this generates a really striking pattern of evolution in the virus that's quite distinctive and that's been puzzling biologists for a while. So we know that there's this very high rate of turnover of, again, hemagglutinin. So the hemagglutinins that are circulating now, none of them would have been around 5 years ago. They've replaced the viruses that were present 5 years ago. And so another way of thinking about this is there's basically this increasing genetic distance with time. And that's what you see here on the left plot. So this is the season. This is 1968 going to 2003. And this is the genetic distance measured by maximum likelihood from one of the strains in 1968. And so again, we see that there's this, again, this fast rate of evolution away from older strains. And that's also what you can see reflected in the phylogeny of the hemagglutinin here on the right. We have strains from 1968 down here and more recent strains up here. And if we were to take this to 2013, you'd see basically the same pattern. So we have this increasing genetic distance with time, and these side branches are very short. So there's a lot of turnover. So this was really puzzling for evolutionary biologists because, or it still is in some ways very puzzling for evolutionary biologists because there's such selection or such selective pressure for antigenic variation. There's such pressure to escape antibody-mediated immunity. And we know, again, that influenza, it's an RNA virus. It has a very high rate of mutation. So why is it that we don't see increasing diversification in time? So some really interesting observations came out in 2004 by Derek Smith and colleagues. They were looking at the antigenic relationships between the strains infecting humans over time. And so antigenic here means that it refers to how the strain is identified by the immune system. And so what they found was that they could group using some sort of, it's like principal component or multidimensional scaling. What they could do is they could group strains from the past several decades into 11 major antigenic phenotypes. And so that's what these kind of blobs, they're called clusters, are. And you can't really see this, but this is basically supposed to be showing you two-dimensional or antigenic space in 2 dimensions. And so what this means is if you're infected with a strain, say, from this cluster, if you get infected with another strain from this cluster, you're probably going to have antibodies that are fairly effective against it. So that means that these strains, even though they might differ genetically from each other, are phenotypically very, very similar. And so this is interesting because this gives us a very different picture of how flu evolves over time. So we're looking again at year down here, 1968 to 2003, but now we're looking at antigenic distance, so what the immune system is ostensibly perceiving. And you can see that instead of this kind of continuous increase in distance with time, which is what we saw for the genetic distance, we see something that's much more discontinuous. And so I was involved in a project that basically kind of took this observation and ran with it as an explanation for how H3N2 evolves over time. So again, what we see are these antigenic clusters that emerge and they replace each other. So there's generally just one at any time. And here we've basically painted them onto the phylogeny again. So you can see there's very little overlap between clusters. So what we realized, and this is it's a little bit subtle, but it has to do with how we parameterize or how influenza models were parameterized up to this point. What we realized is that you can't use the genetic distance as a simple proxy for antigenic distance or as a simple proxy for phenotypic relatedness. In other words, we couldn't simply count up the amino acid similarity between 2 sequences and say, hey, these things are going to have, cross immunity that's directly proportional to that amino acid similarity. There's some sort of discontinuous function. And this is a figure that's basically driving that home. So we have here each of the antigenic clusters that were identified by Smith and colleagues. And over each cluster, we've written the greatest number of amino acid differences between any 2 strains in that cluster. So for example, with Beijing, this is the Beijing 89 cluster, there were 2 strains in here that differed by 10 amino acids. And yet, again, they're considered antigenically to be quite similar. So they're kind of like roughly the same virus. But between these 2 clusters, so these were temporally adjacent clusters. This is the Sichuan 87, well, it's a bad example, but so between these 2 clusters, so the Sichuan 87 to the Beijing 92 cluster, there are 2 strains that differ by only 3 amino acids, and yet they're considered very, very distinct by the immune system. And so this called for a new kind of model. And so the nutshell or the kind of the story that and I'll show you the math in just a second. The basic concept in this model is that there are these neutral networks of genotypes. So a strain is going to start with a certain identity. There are going to be mutations. A lot of these mutations are not going to, again, have any real effect on whether the strain is perceived as different by the immune system. They could cause amino acid substitutions, but they might not. They effectively don't really matter that much. But sooner or later, and we don't exactly know how this works, there's going to be some substitution that suddenly makes the virus unrecognizable or much less recognizable by prevailing antibodies. And then because there's still some sort of cross-immunity between these viruses, that new virus is going to, first it's going to grow really, really rapidly. It's going to expand. It's going to cause an epidemic. It's going to be mutating too. And it's going to displace the old virus. So this is how we argue these clusters emerge and replace each other. They're basically they're these selective sweeps that happen in time. And this is the underlying math. It's basically just a compartmental model. It's like an SAR model, but we are instead tracking susceptibles and infecteds to these different phenotypes. And then we add a little bit of demography. And then we have this cross-immunity function. And this is where we hook up the model to basically like a tunable genotype-phenotype map where we allow evolution of the virus. So how did we know whether this was a decent model for flu? I'm not going to show you all the evidence, but one of the things that this model generates is when a new cluster comes in, and the clusters here are different colors, we get like a spike in cases, which is what you'd expect, right? Because there's a new phenotype, and most hosts aren't recognizing it as well. And then what you also would expect is while there's this kind of neutral or this diffusion in genotype space, you should see an increase in the genetic distance within a cluster over time. And that's what the model predicts. It's also something that we see, though I'm not going to show you that data. But here I'm going back to this observation that when a new cluster emerges, you should get a particularly bad year of flu. So these are real data. This is the percentage of deaths in older people to pneumonia and influenza. And you see that in the years with an asterisk, so these were years when we know there were these major new antigenic types emerging, those were particularly bad years for P&I mortality. So we were pretty excited about this model, and then some new observations came along. And so this is basically looking at that same measure of antigenic distance that I showed you on the previous, or on one of the older slides. So here we're looking at antigenic distance from a strain from 1997. And then here we're looking at time. The different colors correspond to strains collected from different regions. And so you can see that, that obviously there's increasing antigenic distance, increasing antigenic distance with time, but you don't see like super obvious jumps. So this worried us because maybe then our model's not that good. So we went back and we used models now to develop more intuition about what we should be seeing. And so how many people here have taken population genetics? Curious there. Okay, all right, so this is, for those of you who've taken it, this is a Wright-Fisher model that we use. So it's not even a model really of pathogen transmission per se, but we basically allowed there to be kind of these new types that were introduced that were more fit than the old types. And we looked at how that affected certain population genetic measures. So we looked, for example, at how that affects the diversity over time. So this is time here measured in generations. And so when a new fit type comes in, diversity drops all of a sudden, right? Because there's like a new type that's going to start proliferating. And anyway, so diversity drops, and you can measure that as like effective population size, or again, just as diversity of lineages, or this is the time to most recent common ancestor. It doesn't really matter the details here, but you can see that when these new types come in, there should be the sharp reduction of diversity. But then we decided to look at flu, and we don't see that at all, actually. Now, again, this is using data that we didn't have when we developed that model, and we have the new, what are supposed to be the major new antigenic types, that are the different colors, and we don't see these declines in diversity that are well correlated with the emergence of these new phenotypes. So that basically means that epical evolution can't be the whole story. And there are a lot of other hypotheses out there, and the data aren't there yet, and the models aren't there yet to be able to test these rigorously, though people are trying. So other hypotheses could be that the success of a novel hemagglutinin, and this comes back to the epistasis point I made earlier, depends on compensatory mutations or reassortment with just the right neuraminidase, because we know that the functions are quite intertwined. There's also potentially positive selection within clusters. For example, there are these glycosylation sites that flu adds to itself that potentially protect it from antibodies. And then, of course, there are a lot of spatial complications that might distort the signal. And then there are people who argue that that the trajectory of the hemagglutinin actually depends in a very subtle way on how our immune system works, and yeah, it gets pretty esoteric. And then there's also been a really cool hypothesis introduced in the past few years that shows that influenza might not necessarily try to escape antibodies by becoming unrecognizable. What it might try to do instead is actually change its affinity for the host cell receptor. And so instead of, again, shaking off antibodies, it literally just outruns them, and it just binds to the host cell receptor faster. And there are models now that are exploring whether this is potentially a better explanation for what we observe. Okay, so I'm just going to hit a couple other topics in seasonal flu that I think might interest you, and if there are questions, we can handle them right after this talk or in the discussion. So another puzzle is seasonality, and I think you're probably familiar with this general pattern, which is that in temperate regions, the flu is associated with epidemics in the winter. And in more tropical regions, we have sporadic activity year-round. And so this raises, again, an interesting question that I think Dawn mentioned. Why is it that we have flu mostly in the winter in these temperate regions? And this is the subject of a lot of research. One of the major hypotheses is that absolute humidity is driving influenza transmission. So when absolute humidity is low, flu transmission is higher. And this could be through some sort of change in how the virus is dispersed via aerosol. Could have something to do with how well we clear the virus in our lungs. It's not well understood. And then it's been shown for other diseases like measles that there can be, or that there is a very tight association of the timing of epidemics with school terms and the periods when kids are aggregating together in classrooms. And you do see a relationship like that for influenza. And then there's another line of thinking that suggests that maybe it's not so much that transmission is changing, it could be that there's a seasonal change in host susceptibility, and this might be driven by vitamin D. And of course, it's possible that that many of these mechanisms are interacting with each other. So going back, though, to this observation that influenza is, again, kind of causes more abrupt, obvious, like winter outbreaks in temperate latitudes, and kind of has like noisier, less obviously seasonally forced dynamics in tropical and subtropical areas. You do see this for H3N2 specifically, and it has led to some hypotheses about why most of the new strains of H3N2 that ultimately take over, so most of the strains that are evolutionarily successful, why they tend to emerge in East and Southeast Asia. So here I'm not talking about the emergence of pandemic subtypes like H2N2 and H3N2, which were known as the Asian flu and Hong Kong flu. I'm just talking about from year to year when we're looking for, okay, what's going to be the new strain that necessitates an update to the vaccine. They're often strains that we first find in East and Southeast Asia. So a few years ago, a couple groups proposed very similar models that can be lumped into this source sink concept, which is that because flu never goes extinct in East and Southeast Asia, because effectively it's not so seasonally forced, that means there's this metapopulation that's constantly rescuing it from extinction. And this might be why new strains tend to emerge from this region. This is something I'm looking into, but I'm not going to get into the details of that now. First, I'm just going to show you a model that we use to investigate the question, do most new strains of H3N2 actually come from East and Southeast Asia, which is something, again, that most people had just we knew it had basically been a heuristic or an anecdote. So what we did was we took sequences, these are actually full genome sequences that had been temporally and spatially tagged. So we knew the week they had been isolated, and we knew the area in which they had been isolated. So here we're just looking at very, very large spatial resolution or high spatial resolution. We're looking at China, Europe, Japan, Oceania, South America, Southeast Asia, and the United States. So we took, again, these sequences, and we knew when they occurred, and we knew where they were. And we reconstructed a genealogy. So that's what this is. It's not a phylogeny because we have time on the x-axis. And so we were able to reconstruct the tree because flu doesn't, it's really easy to reconstruct flu trees. And then what we did was we used a structured coalescent model to infer what the rates of transition were between these different regions. So how often was a lineage that was in Europe migrating to Asia and vice versa? And we used Markov chain Monte Carlo methods to fit the structured coalescent model. And what we found was, what we got was a matrix of transition rates of influenza lineages, again, just looking at H3N2, and really just the hemagglutinin of H3N2, between these different regions. And so you can see that clearly that there are viruses, for example, or lineages in the United States that will flow to Australia. So it's not that literally everything is coming out of East and Southeast Asia, which is not terribly surprising. But what was important, again, what we're interested in, I should say, is the trunk. Because this is basically showing you where the most evolutionarily successful strain of influenza is. At any time, all the strains of influenza can trace their ancestry back to something that was on the trunk, right? So this is the fittest strain, is the trunk. So we're looking at where the trunk was inferred to be. And what we found is that roughly 2/3 of the time between 1997 and 2006, the trunk was in Asia. And so China here is purple and Southeast Asia is blue. But it looked like there were brief periods when when strains emerged in areas outside of East and Southeast Asia and later fixed. So that means that, in theory, important evolution could be happening elsewhere in the world, and it's not enough simply to focus surveillance on East and Southeast Asia. This result is going to be biased enormously by sampling problems. For example, we don't have any strains at all from Africa, and we have very few from Central America. But it at least suggests that, again, it's not enough to look simply at what's happening in East and Southeast Asia. Okay, so I'm going to talk briefly about other subtypes. Or other types. I've been focusing on H3N2. It is the more common subtype most of the time on average, or I should say more common type or subtype. H1N1 here is in green, and then B is in red, and this is just showing you averaged over time in different regions how much flu is caused by each of these different strains. And what's interesting is there's increasing evidence that there might be interactions between the types and the subtypes. So in temperate populations, it's been observed that seasons are often dominated by H3N2 or H1N1 and B. And there are several immunological mechanisms that have been proposed for these interactions. And as I mentioned, it's an area of very active research. I'm just gonna, this is back to our, you saw these data earlier. This is the deaths to pneumonia and influenza, and the arrows are pointing to years of really big H3N2 outbreaks that are associated with the emergence of these new antigenic clusters. And you can see in the following years, in the years afterward, they were usually dominated by B or H1N1. And you see a similar sort of pattern, at least between H3N2 and B in the 1970s in current St. Petersburg. And you also see this sort of alternation of types and subtypes in the United States. So here, H3 seasons are in green, and H1N1 and B are in red and blue. And so, I mean, why do we care? I think this is interesting because it helps us answer the question, or could help us answer the question, how bad is a season going to be? So this is to give you an idea, again, of like how variable seasons can be. This is the percent of visits for influenza-like illness from year to year just in the United States, and the past season is in bright red. And you can see that year before that, I think, was in green, or 2 years ago? No. Anyway, but there's some years where influenza is barely detectable, and other years where it becomes a huge problem. And so the data that I've shown you suggest that a really good predictive model for influenza might contain information on what the different types and subtypes are doing. And again, potential interaction between them. The degree of antigenic novelty that we're seeing within H3N2, potentially H1N1 and B also, but they don't appear to follow quite the same sort of pattern. We might need to consider multiple components of population-level immunity that are not just specific to the hemagglutinin, potentially neuraminidase, for example. And then we might also need to consider climatic factors and location. And then I haven't touched on this, I think Josh will, but different mixing patterns in different populations, and specifically what children are doing. Okay, I'm just going to talk about pandemic flu very briefly. Your intuition is probably, you probably already know this information, but pandemic flu tends to infect more people and tends to have a higher case fatality rate. And this plot is showing you the what number of deaths we would expect if we were to have the pandemics that we've seen in the past in something like our current population. And so severe seasonal influenza is down here in the bottom left. And these are isocyclines for different numbers of deaths. And so clearly, we wouldn't actually expect, if we were to have the 1918 pandemic now, medical care has been significantly improved, so we wouldn't necessarily expect 1.8 million projected deaths, but it gives you a sense of the enormity of these pandemics. And so the standard answer for why pandemics are usually more severe than seasonal flu is, again, that more people are getting infected, people have less immunity, which could make the pathology more severe, and again, at least in the past, medical care has not been as good. And in the interest of time, I'm going to skip a couple of these slides, and I'm just going to talk very briefly about some of the ways to think about interventions in pandemics. And this is another area where modeling can be immensely useful. So if there's a pandemic, we might think about using a vaccine if one were available. And many times, of course, a vaccine is not immediately available, and so we might think about closing schools, using antiviral drugs to treat infected patients, or we could use antiviral drugs to treat people prophylactically. We could also encourage sick people to stay home. And so a really important question, of course, especially from like a policy perspective, is why shouldn't we do all of these? And assuming limited resources, which ones should we focus on? And so I'm going to give you 4 ways to think about interventions and how we might choose between them. So the first thing to think about is the intervention going to slow the spread of pandemic influenza? And so a lot of different interventions were implemented in 1918, including strict isolation, closing schools and theaters and churches, banning crowding, asking people to use masks. And these interventions definitely had an impact. So here on the left, you're seeing the epidemic in Philadelphia, which implemented no interventions. And the orange curve is showing you the pandemic for St. Louis, where multiple interventions were used. And this little increase here comes from a relaxation of those interventions after several months. So a really critical thing to think about is once you've implemented the intervention, is it going to provide a permanent reduction in transmission, or is the reduction going to be temporary? And will you basically get a resurgence of cases as soon as you stop it? And that's basically what we just saw for St. Louis. But something else that's important to think about is, of course, that these temporary interventions can still be very useful, because one of the problems in pandemics is often that hospitals and medical facilities are completely overwhelmed. And so even if you might not expect to reduce the total number of cases through an intervention, simply spreading them out over time could potentially reduce the case fatality rate. So vaccines are basically, at least in the case of flu, again, unless you're somehow dramatically changing classroom sizes or something like that in a permanent way, vaccines are, for all intents and purposes, the only permanent solution that we have for reducing the size of a pandemic. But then, again, practical questions emerge, like can we afford a vaccine and make one fast enough for enough people? And then can we make an effective one? And 2009, we did make a vaccine, but we didn't really make it in time, at least in the United States. So the red curve here is showing the activity or the epidemic curve for H1N1. And then the blue curve is showing the vaccines delivered as a % of the US population. And if you want to be really pedantic, you might say that this curve should actually be shifted over even further, because after you're vaccinated, it takes a couple weeks to develop a really good antibody response. So we weren't able really to come up with a vaccine in time in 2009, though obviously it has helped. And then another thing to think about, and this is probably not going to be as much of an issue in a couple years because we're shifting how we make flu vaccines, but even if we identify the correct strain, even if we roll out the vaccine in time, the influenza vaccines are still not perfectly effective. And this is really interesting because it's showing data that were collected just a couple months ago. This Perth strain was the dominant antigenic type a couple years ago, and it was replaced. And so all the blue dots here, let's see, they're it's basically showing you the antigenic relationship between strains isolated in the United States between August 2011 and August 2012. And then from the past season, we have these gold strains. And these gold strains, you could say, are roughly centered on this red dot here, which is the Victoria 2011 strain. And this was the strain that was recommended for inclusion in the vaccine this year. This is looking at H3N2. But the influenza vaccine is, currently it's manufactured in eggs. And what can happen is even if you're selecting the right strain sometimes, the mutations can occur on the seed strain, and so what actually comes out in the vaccine can be a little bit different from what you put in, and this is obviously really frustrating. And so what came out this year was actually something that was closer to here. And again, as we move toward manufacturing vaccines by cell culture, this is again probably not going to be as much of a problem, but it just kind of shows you how complicated it can get. And then the very last point that I want to make is that it's also really problematic if a lot of transmission might be happening before people are symptomatic. And so this is showing you R0, so the intrinsic reproductive number on the left as a function of, or plotted against the proportion of infections that occur prior to symptoms or by asymptomatic infection. And so SARS was in some ways a really great disease because it had relatively low R0, and then people were not infectious with SARS until they were symptomatic. And HIV, of course, is potentially more over here in that people can be transmitting it without displaying symptoms. And influenza, this is actually not, flu should actually be down here. So some transmission does occur before people know that they're sick. And this could be potentially quite relevant to the emerging subtype in China right now. There have been 102 cases of H7N9 as of yesterday. It has a very peculiar age and sex distribution in that it seems to be infecting mostly older men. And what's really strange is that roughly 40% of cases have had no known contact with poultry, but contact tracing does not suggest that there's been a lot of human-to-human transmission. So no one's sure exactly how these people are getting sick. And so there's some speculation right now, and there's a lot of talk about modeling. I don't know if there's been much modeling yet. There's a lot of speculation that maybe there's asymptomatic transmission going on that we're somehow not able to pick up. Actually, this is my very last slide. I am starting a lab again in the Department of Ecology and Evolution. I'm going to be studying pathogen competition and evolution. And if you're interested in working with me, please get in touch. Thank you.